Hi everyone, we are going to talk about glycoproteins today. A carbohydrate group can be covalently attached to a protein to form a glycoprotein. Such modifications are not rare, as 50% of the proteome consists of glycoproteins. We will examine three classes of glycoproteins. The first class is simply referred to as glycoproteins. In glycoproteins of this class, the protein constitutes is the largest component by weight. This versatile class plays a variety of biochemical roles. Many glycoproteins are components of cell membranes, where they take part in processes such as cell adhesion and the binding of sperm to eggs. Other glycoproteins are formed by linking carbohydrates to soluble proteins. Many of the proteins secreted from cells are glycolysed, so modified by the attachment of carbohydrates, including most proteins present in the serum components of blood. The second class of glycoproteins comprises the proteoglycans. The protein component of proteoglycans is conjugated to a particular type of polysaccharides called a glycosaminoglycan. Carbohydrates make up a much larger percentage by weight of the proteoglycan compared with simple glycoproteins. Proteoglycans function as structure components and lubricants. So mucins or mucoproteins are like proteoglycans, predominantly carbohydrates, N-acetylgalactosamine is usually the carboxy moiety bound to the protein in mucins. So N-acetylgalactosamine is an example of an amino group sugar, so named because an amino group replaces a hydroxy group. So it is a key component of mucus, serve as lubricants. Glycosylation greatly increases the complexity of the protein. So a given protein with several potential glycosylation sites can have many different glycolysis forms. We call it glycoforms. Each of which can be generated only in a specific cell type or developmental stage. Carbohydrates can be linked to proteins either through asparagine as N-linked or through serine or threonine as O-linked residues. Sugars in glycoproteins are attached either to the amide nitrogen atom in the side chain of asparagine, termed an N-linkage, or to the oxygen atom in the side chain of serine or threonine is termed an O-linkage. Or N-linked oligosaccharides have in common a pentosaccharide core consisting of 3 mannose and 2 N-acetylglucosamine residues. So additional sugars are attached to this core to form the great variety of oligosaccharide patterns found in glycoproteins. Now let's look at a glycoprotein present in the blood serum that has dramatically improved the treatment for anemia, particularly that induced by cancer chemotherapy. The glycoprotein hormone, like erythropoietin, we short for EPO, is secreted by the kidneys and stimulates the production of red blood cells. EPO is composed of 165 amino acids and is N-glycolysed, so at 3 asparagine residues and O-glycolysed on a serine residue. The mature EPO is 40% carbohydrate by weight, and glycosylation enhances the stability of the protein in the blood. Glycolysis protein has only about 10% of the bioactivity of glycolysis form because the protein is rapidly removed from the blood by the kidneys. The availability of recombinant human EPO has greatly aided the treatment of anemias. So, however, some endurance athletes have used the recombinant human EPO to increase the red blood cell count and enhance their oxygen carrying capacity. Drug testing laboratories are able to distinguish some forms of prohibited human recombinant EPO from natural EPO in athletes by detecting differences in their glycosylation patterns through the use of isoelectric focusing. So, glycosylation functions in nutrient sensing. As especially important, a glycosylation reaction is covalent at attachment of N-acetylglucosamine, short for glycan-AC, to serine or threonine residues of cellular proteins. A reaction catalyzed by N-acetylglucosamine transferases. So the concentration of N-acetylglucosamine reflects the active metabolism of carbohydrates, so amino acids and fats, indicating that nutrients are abundant. More than 1,000 proteins are modified by N-acetylglucosamine silation, so including transcription factors and components of signaling pathways. Interestingly, because these glycosylation sites are also potential phosphorylation sites, so the O-glycoenzyme transferase and protein kinase may be involved in crosstalk to modulate one another's signaling activity. Like phosphorylation, glycosamine isolation is reversible, with the enzyme catalyzing the removal of the carbohydrate. So the dysregulation of these transferases have been linked to insulin residues, diabetes, cancer, and neurological pathology. So proteoglycans are important components of cartilage. Among the best characteristic members of this diverse class is the proteoglycan in the extracellular matrix of cartilage. So the proteoglycan, aggrecan, and the protein collagen are key components of collagen. So the triple helix of collagen provides structure and tensile strength, whereas aggrecan serves as a shock absorber. The protein components of aggrecan is a large molecule composed of 2,397 amino acids. The protein has 3 globular domains, and the site of glucosamine glycan attachment is the extended region between globular domains 2 and 3. This linear region contains highly repetitive amino acid sequences, which are sites for the attachment of keratin sulfate and chloride sulfate. Nanomolecules of aggrecan are in turn non-covalently bound through the first globular domain to a very long filament formed by linking together molecules of glycosamine glycan hydrolysis. So water is bound to the glycosamine glycans, attracted by the many negative charges. Aggrecan can cushion compressive forces because the absorbed water enables it to spring back after having been deformed. When pressure is exerted, as when the foot hits the ground while walking, so water is squeezed from glycosamine glycan, crushing the impact. When the pressure is released, the water rebinds. So osteoarthritis, the most common form of arthritis, results when water is lost from proteoglycan with aging, and other forms of this disease can result from the proteolytic degradation of aggrecan and collagen in the cartilage. So another protein like mucins are glycoprotein components of mucus. A third class of glycoprotein we're going to introduce is called the mucoprotein. And in mucus, the protein component is extensively glycosylated to the threonine or serine residues by N-acetylglucosamine. And the mucins are capable of forming large polymeric structures and are common in these secretions. These glycoproteins are synthesized by specialized cells, in gastrointestinal and in other organs here in our body. And these glycoproteins are abundant in cell where they function as lubricants. The defining figure of this glycoprotein is a region of the protein backbone termed the variable number of tandem repeats, VNTR region, which is rich in serine and threonine residues that are all glycolylated. Indeed, the carbohydrate moiety can account for as much as 80% of the molecule by weight. A number of core carbohydrate structures are conjugated to the protein component of this glycoprotein. So protein glycylation takes place in the lumen of the ER or in the Golgi complex. The major pathway for protein glycosylation usually takes place in these two organelles that play central roles in protein trafficking. And the protein is synthesized by ribosome attached to the cytoplasmic phase of the ERM membrane, and the peptide chain is inserted into the lumen of the ER. The N-linked glycosylation begins in the ER and continues in the Golgi complex, whereas the O-linked glycosylation takes place exclusively in the Golgi complex. A large oligosaccharide destined or attached to the asparagine residue of protein is assembled on a different form of phosphate, a specialized lipid molecule located in the ER membrane and containing about 20 isoprene, like carbon 5 units. The terminal phosphate group of this phosphate is the site of attachment of the oligosides. This activated energy-rich form of the oligosaccharides is subsequently transferred to a specific asparagine residue of the growing polypeptide chain by an enzyme located on the luminate side of the ER. Proteins in the lumen of the ER and the ER membrane are transported to the Golgi complex, which is a stack of flattened membrane stacks. So carbohydrate units of glycoproteins are altered and elaborated in the Golgi complex. The O-linked sugar units are fashioned there, and the N-linked sugars arriving from the ER as a component of glycoprotein are modified in many different ways. The Golgi complex is the major sorting center of the cell. Proteins persist from the Golgi complex to lysosomes, secretory granules, or the plasma membranes according to signals encoded within their amino acid sequences and three-dimensional structures. Okay, so that's all for today. Thank you for your listening.